Thank you for the opportunity to talk a little bit about new confocal. It's not new anymore. We actually got it back in 2020, and it was through a shared instrumentation grant with about another 17 faculty members that all participated in order to get it. And I just want to go over some of the highlights of it to tell you some of the things we can do with it. I'll revisit this at the very end. So for those who do not do a lot of microscopy, a lot of the microscopes that we have are fluorescent microscopes that we call wide field microscopy. And the idea of a wide field microscopy or fluorescence microscopy is that you have a light source, usually it used to be a mercury bulb, now it's usually a halogen light source, that shines through and is focused on the objective and emits light and visualizes it. This works really well for some things, but the problem is you can't tell what light is in focus and what light is out of focus. And this gets to be even more of a problem when you look at a thicker specimen. So what confocal microscopy does is it uses a laser, first of all, instead of the halogen or mercury bulb. And secondly, it actually brings the light in, but it has to be able to come back through a pinhole. And that way, the only light that you're going to see is the light that can come through that pinhole, and that's only going to be in focus light. This, for example, is some different pictures on the left-hand side that show the impact of how thick your specimen is as to whether or not you need to use wide field microscopy or confocal microscopy. And in general, the more thin that section is, or the smaller your cell is in terms of its width, the better wide field microscopy is going to work for you. As soon as you start getting a little thicker with cells that are more rounded up perhaps, or histopathology slides or sections, that's when confocal actually becomes very important. The system that we have actually has not just the confocal system though, but something else we call Airyscan, or Zeiss calls Airyscan. And what happens here is we can use two different modes for the confocal system that we have. One is where the light has to go through this pinhole, and you'll get just the focus light that way. But they also have a new system that you can open that pinhole and send it to the Airyscan mode, and there's 32 different detectors that actually get light in focus. And what that means is you have an increase in the signal to noise ratio or sensitivity, approximately tenfold. And it also allows you to look at thicker sections than you can with confocal alone. So we can use both of those modes in the system that we have. So each of the 32 detectors on the Airyscan collects additional information and creates more light. And in using the 63x oil objective, the microscope resolves structures that are about 120 nanometers apart in the lateral dimension and 350 nanometers apart in the axial dimension. So that gets us close to the range of what they call super resolution microscopy, but just using regular fluorescent staining. So it gives you the enhanced signal to noise ratio, improved resolution of spectral data, more spatial information, and importantly, it works better for thicker sections than just a confocal alone. I'm sure you've talked a little bit about the two-photon microscope. That's even better and more necessary for thick sections in animals. But the Airyscan helps bridge a little bit of that gap. This is a picture over here on the left-hand side that shows HeLa cells that are stained with the confocal here and the Airyscan here. And you can see that you do see an increase in resolution. You don't see as much of that increase in signal to noise ratio here. I like small protozoal parasites, single cell parasites. And so this is a picture here of Toxoplasma gondii. The red here are actually, each one of these is an individual parasite, and they grow in this thing called rosettes inside of a cell. And we were particularly interested in something called a Fic kinase, which is a novel kinase that's localized to the basal end of the parasite and is only found in apicomplexan parasites like Plasmodium and Toxoplasma. So with a regular wide field, let me back up. These are really small cells, so usually wide field fluorescence microscopy works really well for that. And you can see here, we can actually see the staining of the, in green, with that Fic YFP kinase in the rosette where the parasite's replicating here. But when you bring it to the Airyscan, you can see now that actually there are, instead of just seeing the green here, you see that there's two juxtapositioned right next to each other that may make a pore or something like that when you do a cross section. So that gives you an idea of some of the difference in resolution that you'd see with a wide field microscopy versus confocal or the Airyscan. This is just a picture here of plasmodium and red blood cells. This is also a gentler way of imaging because you're not using as much laser strength, and so it's good for live cell microscopy. The other thing you can do with the system is 3D reconstruction of Z-stacks and XY tiles. Z-stacks are up and down through the density of something. XY is side by side. And this gives you an idea of what this looks like in practice. This is an optical sectioning in Z and XY planes of a pancreatic islet specimen from a patient with recent onset diabetes. And you can see just here the insulin is shown in the green, the pre-insulin in red, and the glucagon is shown in blue. And what this allows you to do is if you had a section of an organ or a tissue, you can do XY scanning to get the entire breadth of that tissue and Z to get the entire depth, and then put everything back together, stitch all the different pieces together, which will be done automatically, and then do a 3D reconstruction of it. This is a picture here of a screen that was done looking at breast cancer cells. So they actually looked at breast cancer tumors in mice, and they had a drug that they were trying to figure out how it worked. And what they were able to do is they could, I screwed this up too there. They could visualize the endoplasmic reticulum, as shown in red, in all of these different tumor cells in this tumor. And they learned that actually the drug was acting by disrupting that ER and causing ER stress, and that's what was killing the cancer cells. But that's just another idea of that 3D reconstruction that you're able to do. Microbiome. So this is a picture here of different microbes that are on the surface of somebody's tongue. So again, you can look at the overall structure and the different type of bacteria and the microbiome itself in different locations. Another thing that you can do with the 3D imaging of tissues is when you image the entire section, it will take a map of that entire section, and then you can tell it where you want it to zoom into in terms of point out this location. It'll document that location, show you in high resolution with the 63x objective what it looks like at high resolution, and then you can go back to it again because it can remember where it was located. So it forms that map, and then you can actually dig down once you find the spot that you're interested in. Another area, this is an inverted microscope that we have, and what that allows us to do is multi-position time-lapse imaging. So we have an environmental incubator with the system, and automatic and definitive focus. It has CO2 and temperature that's regulated. And in theory, I can put my nice little parasites up there at the top, right here and here, along with fibroblast cells, and take a picture every, say, 15 minutes for 24 hours, 48 hours. And then I can actually visualize that sequence of events. So here are two parasites. The green shows the Fic kinase in the basal end of the parasite. They're invading into the host cell. At this point, they've replicated to 2 parasites per vacuole. And here they're that rosette showing that the Fic kinase there. That's the parasite, that's what I work with, but most people work with other cell types, and you can do that for human lung epithelial cells or any other viable cell type that you're interested in. This is a zebrafish. So you can do the same type of things with something the size of a zebrafish and looking at live cell zebrafish without disrupting them. And again, you can get an overall picture of your fluorescence in the zebrafish. And then as you see the squares here, zoom into those specific areas that what you want to look at. And if you could we're actually doing this by time-lapse microscopy, create a video of this, and then actually present that video. It's also used for something called FRAP analysis. This is fluorescence recovery after photobleaching, and it's particularly important for kinetic analysis of diffusion trafficking through cells and tissues. What you do is you shine an intense, very narrow light in your area of interest, and you see how long it actually takes for the label that you're interested in to come back to that location. And you can actually break it up into different parts and see the trafficking pathway of how it actually gets there with live tissues or cells. You can also do FRET analysis. What FRET analysis is, it looks at molecular interactions. You have two molecules, both with a different fluorochrome, and when the two come together, they fluoresce a different color. And that will tell you that you're having, that they're interacting with each other. And so you can do FRET analysis with the system as well. One thing that some people have done with it that I would have liked to have when I was a postdoc is you can do live cell screening of your system as well. So you can take a 96 well plate, and let's say again I wanted to see what happened to my parasite and macrophages. I could do mutagenesis of the parasite, come up with 6,000 different mutants, put them on my plates, and screen them and have it automated screen to see where they have a defect and where they don't, and then go back and pick that population up and then take that out and study it. We also have, this is an inverted microscope. The one reason we went with an inverted system is it allows us to look at live cell imaging on plates and tissue plates and 96 well plates and other types of Petri dishes. I know a lot of people though like to actually be able to dip their objective in from the top. And so we have something called an LSM Tech objective inverter that it can be placed within the microscope system. So instead of acting like an inverted microscope, it'll come from the top and be more like an upright microscope. So that's covering a lot of territory very fast. I wanted to give you a brief overview of some of the things we can do with it. If I didn't cover something that you think it could do, come and see me, set up an appointment with me, and we can talk about it and see what we can do. But this is a system here, and special thanks to the school for supporting the NIH grant that we put in and all the faculty members that participated in it. Thank you.
Thank you so much, Dr. Mordue. We're running a little bit late. Yep. Do we have any questions? We have time for maybe one.
So Dana, you showed this really interesting picture of the rosettes of the toxo.
Yeah.
And so at one point you showed a sort of blurry picture on the left and then a higher resolution picture on the right. Was that the same field taken with the Airyscan on the right and the...
I don't know that it was the same field. It would be the same type of rosette.
Yeah.
I think it was actually the same field. I'm not positive, but I think I actually did a demo over with Zeiss, and they were showing off their Airyscan, and so we had the same one and we looked at it again, but I'm not positive about that.
Yeah, no,
we tried to match it up, but
yeah,
I'm not sure.
I see. Because I mean, it looked remarkably different.
They were remarkably different.
So
Yeah,
you didn't see all that spread of the GFP.
But remember, that was more wide field microscopy, so it would not have actually been the same.
Oh, that was wide.
Yeah, so that wasn't confocal, that was Airyscan. Okay. So that would be what you'd see with wide fields, yeah. So
during that period, we also went to Zeiss, and
I thought about using your pictures.
We had a couple of pictures with Airyscan and not Airyscan on the same.
It can make a difference. Where it seems to make the most difference using the Airyscan mode is if you have, first of all, a thicker section that you're using, because the Airyscan actually buys us a little bit more in terms of distance, in terms of being able to penetrate. Or if your signal to noise ratio is low, it's really good for that in terms of amplifying that.